Hello, Stefan.
Hi, Johannes.
Cool to be here.
Yeah.
Yeah, happy to have this Smart Lab webinar. I thought this time we make it the two of us as sort of the New Year entertainment, probably a bit better than looking at slides and only one person speaking. Really glad you're here.
And thanks for inviting me.
Yeah. And what we want to do today is to share a little bit our insights, how you can implement lab automation into your education. Stefan has great experience there with our bachelor students mostly. I'm doing some lectures in the master courses, and we both have students already in master thesis, bachelor thesis, and industry. So Stefan, maybe we start first with a quick introduction
Sure.
of ourselves. So could you introduce yourself, a little bit your background, where you come from?
Happy to do that. Absolutely. So, as you said, my name is Stefan. I actually studied here at the Fachhochschule Nordwestschweiz, my Bachelor and Master studies a few years back.
Wow.
And I was focusing on organic chemistry during my Bachelor work and thesis. And afterwards, the Professor Götz Schlotterbeck started, and he really convinced me to go into analytics. And this is also where my journey for lab automation started, with the robot, which we developed back then. Which is actually this one. I just realized.
Yeah, come on. Yeah, we're sitting here in the middle of our automated lab.
Here the journey started. I continued my master studies with analytics, did a lot of automation. Then I quickly did a detour to go to teaching. So I was teacher for the apprentice of Roche for half a year. And then I was hired by Kamok to really develop and promote this piece of equipment, which is auto sampler for dry blood spot cards. So I did some product management for this system. I did a lot of analytics and lab automation. And as the business grew, I was also doing a PhD on this very topic to automate diagnostic tools from the liquid blood analysis to a miniaturized version, the dry blood spots, which we then were able to fully automate the whole pre-analytical process. And then business development and so on. And yeah, a little bit more than 2 years ago, I made the change back to academics and was given the position here as a professor for instrumental analytics, where I do a lot of mass spec and lab automation as well. So this is a little bit my background. Yeah.
And your career. Cool, exciting. And diagnostics, dry blood spots, I think this is a future technology that will revolutionize as well the diagnostic world.
And we have quite some expertise in our team with this and are really building on this and are quite excited for the future. So Johannes, everybody knows you, or do you also want to say a few words about yourself?
Yeah, I'll probably say some words as well. So I'm with Tim already sort of since the beginning of the Smart Lab network, but first time here in one of the webinars. My name is Johannes Mosbacher. I studied actually physics, did my PhD then at the Max Planck Institute in Heidelberg. And there already having sort of dinosaur, old-aged pipetting tools that were automated, programmed with modular tool software. I don't think that anybody uses this language anymore. Then, after a postdoc at EMBL, joining Ciba-Geigy, turned into Novartis, where I was also responsible for building assays that came into high throughput screening. So from manual pipetting up to the automated high throughput screening equipments. This I did quite a long time, for 12 years at Novartis, then moved to Actelion. Also a white startup company built from Roche spin out for 5 years, where I'm also doing a lot of high throughput equipment, sort of using functional imaging, high content imaging, but also calcium imaging machines and a bit of automated electrophysiology. And then moved to Roche for 7 years. And there I was also in contact again with the high throughput screening department, but then also learning with Martin Strahm, for example, and colleagues from PRED-I, how an electronic lab notebook is installed and how this is optimized. And then since 5 years, I'm now here at the Fachhochschule Nordwestschweiz as a professor for pharmaceuticals and still running a lab with in vitro biology assays. And I'm also responsible now for a master's course in lab automation. Yeah, that's basically it. And we are both super interested and motivated to bring these lab automation expertises to our students. Because their lab environment, when they go to their jobs in future, they look already quite automated, right?
Absolutely. I mean, as you can see in the background, I mean, the analytical lab especially, there's a lot of automation.
Yeah, and I think also cell culture, pipetting, this is today mostly in our industries. We have the big brands here, Novartis and Roche, but also a lot of startups and smaller companies that use now liquid handling robots. They use electronic lab notebooks. They use digitized data sampling and diagnostics. So it's really necessary to get them on speed with all these automation tasks. Yeah. Is there anything special in diagnostics where you think lab automation brings in companies like VOE a lot of benefits?
I mean, there are several aspects to it, I think. But I always like make the connection between lab automation and digitalization because one without the other is a little bit not working. And I think especially for diagnostics, you have the digitalization. That means you track your samples very precisely. So, for example, you get the patient data, the information, linked to some sort of a barcode, QR code, RFID, whatever that is connected to the sample. And then you have this connection that somehow stays all over the process and that you can really trace your data. And this needs to be somehow in a safe environment, in a healthcare cloud, because you don't want to have the patient data spread out to the network.
Oh, true.
And so all the diagnostic tools, the equipment we have along the way need to be compatible with this labeling system somehow. And so this is really something integrated. And then you have the lab automation aspects, which is clearly, I mean, if you go for high throughput, there are many tests and there's a lot of repetitive work that you don't want to pipette yourself.
Indeed.
And but there's also safety. I mean, one aspect is also safety. I mean, there's biohazard too, if you have blood, for example. And if it's just in a sealed environment, you can easily have this biosafety 2 or even higher regulations because only the machines are interacting with the process and there's not much coming in and out. And the other thing is our instruments, they get more and more sensitive. This is amazing. Just during the last 10 years, mass spec, how much more sensitive we got. This is just amazing to follow up. And then if we see diagnostics, I mean, we take a lot of blood today. We take venous blood, 5 ml, to do a lot of diagnostics. And we do this since many years, and at these years, the instruments got more and more sensitive. So in terms of taking a look at our resources, I mean, green chemistry, etc., we can miniaturize everything. So we can miniaturize the blood drawing. So instead of the vein, we can go capillary. We can miniaturize the device where we collect the blood, the container, which we ship. So we use less plastic or cotton or whatever this devices may look like. And within the lab, we need much less solvents or buffers, and we can streamline our automation to handle this task. And if we go below microliter, like ranges, I mean, you know how it is to pipette a microliter. This is just not precise. But our robots, they can do it with highest precision. So I think these are 3 main aspects. So the cost side, the safety side, and the precision in very small volumes that are a key driver for this field here in diagnostics.
Yeah, indeed. So in my field, sort of pharma and mostly research in pharma, I may add, of course, the sheer throughput. The pharma companies have millions of compounds in their library, and to test all of them against a target in a sophisticated, maybe cellular assay, you need automation. We did once a by-hand screen back at Novartis 20 years ago. on the GABA B receptor once it was cloned. And that was sort of, I think we got 2,000 compounds. It was an effort of 5 people working the lab day and night to get this done. And nowadays, you cannot imagine to do this anymore. You have your pipetting robots, you have your plate handlers, your automated plate hotels with the incubators. You need this automation to get all the compounds tested in screens.
But Johannes, what about the data? I mean,
exactly.
if you have this huge amount of tests,
true.
What do you do with the data? How do you handle it? Do you need data cleaning? Or do you have the same result from plate 1 to plate 200?
Yeah, not at all. And I was super amazed by Thomas Sander's group at Actelion. When I came from Novartis, which has always this backlog of data, so they wanted to be compatible with data that was generated in the 60s, 1960. And it was really always a problem to get the database consolidated in a way that you can use it, that you can compare 2 graphs one by one. Thomas Sander at Actelion, of course, they could start from scratch. And together with Oliver Peter and the high throughput screening team, they built everything from scratch. You could super nicely visualize data. I think the data suite is still there out in the public, and you can use it. And that was the first time I've seen what is possible. All Java-based scripts, really nice. So making filters, correcting some plate errors that came in, visualizing the data, that you could sort of look at all the plates at once in a sort of movie. That was cool. Yeah. Yeah. And even before having then the hardware all automated. So I agree, data automation and processing, this is really also super important.
That doesn't sound like an easy task.
And it's also difficult to teach students, right? We are here, and I think you remember, we still have, when you studied, we probably still had these paper pen lab notebooks. We still have one practicum here teaching paper pen lab notebooks that the students have to write, handwrite their experiments. And then moving on to a fully organized electronic lab notebook. That's a major.
And maybe soon you just tell it the notebook.
Right.
We'll see.
I was always surprised about companies like Eppendorf, Chilson, that they have manual pipettes, that you cannot do what you always do when you're in the car saying, please call my wife, please tell my daughter this. And you cannot go to a pipette and say, please pipette 200 microliters, and it's setting itself to 200 microliters. Why is that not possible yet? These are smart lab solutions that I'm wishing to have in 5 years.
Yes.
Yeah.
And these are smart, like ISIL solution?
Sure.
Or island solutions?
Yeah.
Which I think is really the way to go forward. Before connecting everything, we should have like smart islands and then start interfacing these so-called islands, I think.
Yeah, true. And there is something really fundamental to that. Maybe 10 or 5% of the jobs that our students will get when they are ready here will be in fully automated environments. Most of the students will be in these partly automated lab environments where these island solutions make much more sense. A balance, solving some of the ingredients, making a dry blood spot, something like that in a diagnostic lab, handling an ELISA kit, for example. And they should learn it there as well. And then they can move on to these fully automated and integrated lab automation solutions.
I agree.
Yeah.
I would even go one step back. They should first learn the science to understand the process, and then learn to partially automate before they bringing the things together. But I think there's also a big challenge with increasing automation is that we lose the knowledge how it actually worked in the beginning.
True.
I mean, and this is crucial for understanding the process and being able to optimize.
Yeah, absolutely right. And I think this is why we have in our education, in the practical education, which we have here at Fachhochschule, we have always a practicum where they learn everything sort of handmade from scratch. And then they implement tools like QR codes on the bottles to read, oh, this was a buffer that was done by the team last week. And they got good results, so we trust this buffer, we can use it. Or an electronic lab notebook that they can learn, oh, I don't need to write again what I did because I can copy paste because I did it already before. I save time.
Absolutely.
Right. Yeah.
Speaking about education, maybe you want to give a quick overview about what we do here?
Oh, yeah, sure. Yeah.
To come back to the script.
Do we have a script?
Don't tell anybody.
Right. Yeah. Fachhochschule Nordwestschweiz, sort of University of Applied Sciences in the larger Basel area. We have a School of Life Sciences, and we are in the School of Life Sciences here, both of us. And the School of Life Sciences has 4 institutes. Institute for Chemistry and Biology, Bioanalytics.
Bioanalytics, yes.
ICB, Chemistry and Bioanalytics. IPT, where I am, Pharma Technology. We have IEC, Ecopreneurs, for environmental research. And we have IM2, which is Institute for Medical Technology and Medical Informatics. Medical informatics and medical technology is also highly automated, but we won't touch this today because I don't have the background, frankly. And also the ecopreneurs we don't touch. We are more in these labs, which are diagnostics, analytical, and pharma labs. This is mostly what we cover. And there we've built in the last 3 years a curriculum in our education. Stefan is more in the bachelor student education. I'm for the master's lab automation responsible. Our masters start actually quite heterogeneous. So they're coming from different universities with different backgrounds. And we only have theoretical courses. So we have one course, lab automation, where we teach them the basics. So actually, how is a lab running? And then what are the elements that could be automated? And then how to do it. So what is the need to automate? And is there always a need to automate? If there is a need identified, how to do this? What tools are around? And then what is for me very important to also have KPIs, key performance indicators that tell you this automation really makes sense and is better than what you did before to really show it made sense. And then having case studies where the students then learn. So you gave also lectures there where you give the case study of blood sample diagnostics. We went to VOE, we went to Roche QC labs to look a little bit, how do they do it? And then the students learn what are the applications there. Master is more what I said theoretically, but they apply it then in their master thesis, which runs for 8-9 months. And mostly in our industry partners, one example is a student that we currently have at Roche PRED, trying to automate for pharma applications and other labs. So this is a bit what we do in the automation. We also have more practical work in the bachelor curriculum, and Stefan can tell you a little bit more about what you're doing.
So I would say the lab automation in the bachelor nowadays it's really omnipresent. We try to integrate it everywhere. So we have dedicated modules. One module is lab automation and digitalization, which we teach in the bachelor for 5th semester students. So they're already quite advanced in their bachelor studies, where we really take a look at what can we automate and does it make sense to automate?
Right.
Because if you have a bad process automated, it's still a bad process. So, so therefore, does it make sense and is it feasible? If it is feasible, what tools do I have? What do I have? And do I need to adapt these tools? So, for example, I have a robot with 6 axes. Maybe I need to 3D print a new part that it can pick my dedicated sample, for example.
Right. Yeah.
Or I need to modify the code. So I need some knowledge about coding. And so on. So this is really a lecture where we are half in the lecture room and half in the lab. So we really have like a lot of practical things that we integrate, and we have practical courses where the students are also faced with automation challenges.
Yeah, I think the bachelor in our school it's about 10 weeks of theoretical lectures and then 6 weeks of practical courses in one semester. So there is really a good balance between first learning the theory and then applying the theory in practical ways, like biochemistry, practicum diagnostics, bioanalytics, pharma technology, practical work.
Yeah, and from my point of view, this is really the essence of our education. Because I always say there's university, there's industry, and we are somehow in between. Yeah? So we learn the academic knowledge. Yeah, maybe we don't go as deep as the university goes, but we have a lot of lab work. So we really apply. We apply, and then we learn, we iterate, and our students, they can really go to the lab and know what to do. They know how to manage projects, do the time planning, and they know how to work with the equipment and also with the automation.
Yeah, this is the big difference I experienced when our students go into an industry partner's lab to do the bachelor or master thesis, that they come back and say, oh, it's so amazing. Your students know how to calibrate a pH meter, know how to run an ELISA kit immediately, run a PCR, run an bioanalytics, HPLC. They sit in front of machines and don't break it, which is different to, for example, what I did when I started as a PhD student, even at Max Planck Institute, where I didn't know anything coming from university. You only learn theory, but not the practical handling. Right. Yeah.
Yeah, but on top of that, they also know what is a design of experiment. How can I optimize parameters in a scientific proven way, and bring this scientific background and data skills together. I think is a very powerful combination.
Yeah. Yeah. Indeed. Again, with this notion that lab automation itself is not a real basic science. It's not like physics where you can have laws. It's not like a chemistry lecture where you have sort of compound X plus compound B gives a reaction and then compound C. It's really more an approach. It's sort of a trade know-how.
And I think, I don't know how, if we can translate this saying, German saying to English, but there are several routes to Rome.
Exactly.
I don't know if this exists.
Right.
But,
yeah.
I mean,
certainly.
there's also like several ways to solve automated sample prep, for example, with different equipment in different ways to reach the same goal. I mean, this is also
that's true.
Therefore, it's not a hard science. Like,
exactly. Yeah. And we always are surprised each time the students have a challenge, solve it, and come with solutions that we never thought of. Yeah. I have one start with my lecture in the masters. This is probably the only practical thing. I give them electronic pipettes, multi-pipettes, and then a 96 well plate, and then buffer and ink, and say, please pipette a checkerboard layout. So buffer, ink, buffer, ink, buffer, ink in all rows. It looks like a checkerboard, and come up with the smartest solution. And smart can be fast, can be resource sensitive, can be the least steps to do. And they always come up with something new.
That's cool.
Really surprising. Yeah. And we realized as well, a liquid handling robot takes more time than a person pipetting it for one time. If we need it 100 times, automation makes sense. If it only
so if you include that you need to program the robot.
Exactly. Yeah. Also, I think the timing last time, the students even stopped the time. And one student was even faster than the liquid handling. But the liquid handling robot wasn't done on full speed because we are always cautious not to break all the...
No, but I can imagine.
All the arms. Yeah.
I mean, also extracting the dry blood spots, if you do it manually, maybe you're a little bit faster.
Yeah.
But you don't want to do it for like 2,000 times in a row. I mean,
exactly.
This is also a little bit...
Yeah. And telling them that you are faster done with your thesis work if you use the night times in the lab. So if you can run a robot overnight, they are 24 hours 7, whereas you hopefully only work 8 hours, maybe less.
Yeah, we recently had a thesis in our lab where we developed a sample prep for MALDI, matrix assisted laser desorption ionization, mass spec. And that was an amazing work. And the student really came up with approaches which we didn't think of in the beginning. And I think this was a quite nice example. I actually made a video of this thesis. Maybe, I don't know,
cool.
if
Yeah,
we can,
Tim could start the video
maybe
probably now.
That would be good.
Yeah. Otherwise, you find it at the end of our video clip.
Or we link it to the show notes.
Exactly.
I don't know if you have show notes.
Like what the YouTube stars always tell the influencers, see my QR code below. But that's I've watched it and it's really cool.
This is really like, and this is something they're capable of when they finish their studies here.
Right.
Yeah, they understand the science, they can do it, they can program it, they develop concepts to even make it smarter and more efficient. And now he was just like, he found a job within just a few weeks at Chemspeed, and he's now like going to the next step to do like the full automated like synthesis platforms and so on. Yeah, this is really some future skills, I think, which is required from the job market, that we can like educate here with our tools.
Yeah, that's really cool. And Stefan, I super appreciate it because you're already sort of a digital native. I guess I'm not, with the gray hairs.
Maybe more than you, but there was no smartphone when I grew up. So I still had to call my friends' parents to ask if I can see them.
With the line phone, with dialing. Yeah. That's right. So it's also a challenge for people like me who went through all these changes to teach the digital natives to say, why is it not working like this, or try to think like that. We are also always surprised about their approaches and their sort of background and how they actually attack problems. I didn't find out completely how to best connect with them and with their former skills, but we are learning as well. So I always have in the first lecture in the semester one scheme where there is an airplane, but it's still on the way to be built. So the airplane is already flying, but there are still people fixing something on the wings. The back is not yet fully done. We are already flying with education, but we are always adapting. And always
what I observe a little bit is also like, it goes back to Darwin. I mean, survival of the fittest. And I think there are some students that are super quick adapters to new technology. I mean, there's a new tool out. Next day, they know it. 2 days afterwards, they apply it. And this makes them super efficient, super smart. And this is something which I observe with pleasure, because I actually don't have this time to screen all the tools and try to understand them in that way.
Yeah, true.
All the time.
And it's the same with me. So my programming skills are sort of former times, modular 2, I mentioned, C++, other things that are not existing anymore today. And nowadays you can go to ChatGPT or other LLMs and say, please code me this thing. And there's an R script or a Python script coming out. So I think the way how they will in future actually handle lab automation might be very different to how we did it. But we need to, at least what you already said, teach the basics.
Absolutely.
The concepts. So
that's the foundation.
When is it necessary? What is actually possible to do? And then can I measure that I have really also success?
Yes, absolutely. To understand the principles and also how to validate the system, that you not just create a black box. So maybe your black box works, but it comes to the right solution with the wrong input. I don't know. And then it works for somehow, but you don't know.
Right.
So you somehow need to understand the principles, or also how to validate the whole process.
So we in the masters also always do teamwork, that teams of 2 or 3 or 4 students present to the others. And sometimes there is a coding teamwork in where we give them a certain little challenge on a liquid handling robot. Say, please take samples, for example, for bioanalytics, take each time samples from a 96-well plate, assuming, for example, an atomy assay, which is important in pharmacokinetics, and then pipette this from a 96-well plate to a 384-well plate, seal it and move it to the autosampler for the LC-MS. And so I think from these case studies, also the other students learn then, aha, they approach it like this. Might be I have an idea to do it differently. And then they are curious to also try it out. So having these case studies is, I think, also a good way, because there are no really common rules. There's not like a knowledge that you can sort of from top down teach. It's more bottom-up, case study driven and application driven.
I agree. I remember myself back during the studies, the case studies which we had to develop. I still remember these examples.
Okay.
So this is really something where I think you make some kind of a big learning step and apply the knowledge or condense it. Or,
right. Yeah, interesting. I think in the Bachelor, there will be also new things coming into the lab automation, a practical course. Do you know a little bit more about that?
So we had a big discussion if we should have like lab automation practica. And then we discussed several options. So we have dedicated practica for lab automation, or we integrate lab automation in our existing practica. And basically we couldn't decide. So we have both now.
Okay.
So there's colleagues from bioanalytics. They do a dedicated lab automation practica where they have like different modules. And then 2 days they do 6-axis robots. 2 days they do XYZ robots. 2 days they do excursion to Hamilton and so on. And I decided the other way.
Okay.
because we have so much automation on our GCMS, LCMS sample prep. Like it starts with the autosample. I mean, automated injections. This is like so common since, I don't know, 30 years now. And these autosamples, they went along the line to sample preppers. And now they become fully automated workstations from A to Z. And a little bit this evolution, we have several techniques here in our lab. And I mean, the students, they're confronted with these systems anyhow, because during their education. And so we especially give them, after they visited this lab automation lecture, we give them extra tasks to automate it, a little piece of their workflow they develop in their final practical work here, which is just before the bachelor thesis. And then we have several projects in bachelor thesis which are really like focused on lab automation or digitalization. So this is a little bit how we handled it, because we think they're already quite advanced, and they should bring it together. And with analytical chemistry, it was a little bit more streamlined than with the bioanalytics, because there it was just like much broader. Than here we have like on the GCMS with this CTC PAL systems or Gerstel systems that can automate like liquids or also from solids directly into the GC. Same on the LCMS, moldy TOF as we just heard. Or other sample preppers as the Hamilton instrument we have here in the background as well, where we can do like whole liquids sample preparation workflows.
Yeah, it's cool. And I have to add something on the bachelor, because I'm also running a bachelor practicum for earlier semesters, so starting first semester, then second semester. We already start there with little tasks, isolated, like what you said. This is also important. Start small, but tell them already what is digitizing lab work. For example, how do you track a sample? This is important for blood samples. You mentioned it already. For the Humanforschungsgesetz in Switzerland. You need to know where your samples are. So how is it actually to code a sample and then put it somewhere and then trace it in your notebook and see you can actually find it back? And also to use an electronic lab notebook, where we use OpenBIS right now, developed by ETH Zurich, for example, where we teach them in simple ways, isolated sheets, how to actually put an ELISA assay or a pharmaceutical assay into an electronic notebook. And then they move on and learn things like that. So I think it's bits and pieces from the start on, and then accumulating in the 5th semester, like what you just said, and then apply it.
And then sending them back to you to the master course.
Exactly, yeah.
To do
where
the
the
final
master
finish.
exactly where the master course really goes deeper inside and having probably a bit more coding. We now also have a new curriculum on pharma informatics. Nahum just started that last year and quite successful now. The students come usually with either an informatic background or very IT aficionados. And they then really go into coding, taking challenges from normal lab work and say, how can I actually program that, digitize it, automate it? And I think this is also very fruitful in the masters to have different backgrounds together.
Yes.
The students learn so nicely peer-to-peer. We don't need always to interact because they also cross-pollinate each other. IT students train other students how to code. Other students tell sort of pipetting students tell others, but you need to watch that, for example, acetonitrile. If you pipette it and it warms up, actually it doesn't behave like water, which maybe an IT guru never experienced in his life, in her life. So this is also important.
But I think this is also really the skills they need for the job afterwards, because in the team, you know it.
True.
That exactly. It's never just like chemists or biologists. It's always like working together with other specialists and then speaking the same language as they are. We just had the discussion that a validation is something completely different for me than for my colleague from bioanalytics.
Oh yeah, yeah. And very important. And for regulators, really, again, very different.
Exactly. So this is really like something I appreciate here in our education, that we also bring the different skill sets in the master together and bring them to interact. And this is a lot of also soft skills they learn next to the hard science from the books.
Right, yeah. It rings one bell, sort of this regulation and validation. We don't have GMP or GLP labs here at our school, luckily. But we also teach a little bit the benefits of automated lab environments for those regulated experiments. To say, why do you need to always have a 4i principle when pipetting something if you have a liquid handling robot which has an audit trail and where you can read out, okay, this robot pipetted at this time in the night, maybe this amount of volume.
Or even a balance. So we have close collaboration with Mettler Toledo. And then we have a balance, an automated balance, which does the weighing itself. So it's absolutely amazing technology. And they're actually coming for 2 hours to our lecture during the bachelor, and they teach us data integrity.
Oh yeah, yeah.
So why do we need data integrity? Why do we need, like, as you just said, audit trails, etc.? This is especially for GMP environments, but also for non-GMP environments. If you do like research,
right?
I mean, it's also good if you have your Heureka moment, to know what you did before.
Exactly, it's crucial. Be able to reproduce it.
Yes, exactly. And this is I really appreciate this partnership with Mettler to have this strong knowledge for building actually GMP processes.
Yeah. That's right. We can have a lot of help from outside. This is really good. And
that's
also
amazing.
The smartlab.network can actually help with their expertise. So we also have guest lectures. We have site visits, and every company here in the Basel area is happily invited to contact us if they offer some site visits for our students or bachelor internships, master internships. International placements are also super interesting. So our bachelor students sometimes go abroad to learn. They want to know what is going on in Europe. How is it handled there? Because of course, it's a Swiss environment. We have many of our students ending up in a Swiss workplace, but they are sometimes also going to international labs, international companies. And super important. Also for research collaborations, FHNW, School of Life Sciences has a lot of EU consortia collaborations. And we are always looking for more international exchange, especially when now Switzerland is back to EU Horizon Europe programs.
Yeah, I think science is a collaborative thing to do. Also on academic level, I think we should interact and learn from each other. That this brings us further. For lab automation, I think this is really like a big topic. As we just discussed, there are several ways to lead to the same solution.
True.
And if we discuss and exchange regularly, not just in the Swiss environment, I think this is super powerful.
Exactly. So happy to learn about all your experiences, your expertise in the Smart Lab network. And I think we conclude now. We hopefully entertained and informed you a little bit about our approach to teaching lab automation. And then back to Tim, I would say. Yeah, thanks a lot, Stefan.
Yeah,
that was really great. Thanks, everyone. And then, Tim, your turn.
Wie lange haben wir jetzt geschnurrt?
Wahrscheinlich schon sehr lange.
Ja, ich weiß, viel kriegen ist